Welcome to Chapter 15, the Adaptive and Specific Immunity. We will end this chapter by discussing distinct immunizations and how those vaccines are prepared to provide efficacy. So our specific defenses are indeed those third-line defenses. They include our B and T cells. They allow us to be protected against things such as cancer, as well as these protect us if our non-specific defenses fail. The non-specific defenses were covered in Chapter 14. The adaptive immunity consists of a dual system of B and T cells. B and T cells both have to recognize antigens. These are molecules that stimulate a T cell response and a B cell response. In particular, the antigen will have distinct receptors that we'll discuss a bit later that are specific for that antigen and necessary for recognition. Because of the specificity, we have distinct antibodies that will function only against that distinct antigen in which it was in response to. Additionally, our cells can develop memory responses to where we have T cells or B cells that will immediately lyse or remove an infectious agent if we recall it for a second exposure. The development of immune response, first and foremost, is to perceive and attach to non-self or foreign molecules. We have to, of course, tolerate self molecules, as well as we then have to commit chemical signals between these cells to get them activated. And so once we have this process, these cells will then develop or differentiate into distinct lineages that are specific for the type of infection they have encountered. Part of this recognition process is that of the receptors on the surface of the cells. So the antigen-presenting cells, or our host cells, have MHC class 1 or class 2 receptors. All cells within the body, except for red blood cells, possess an MHC class 1 molecule. This MHC class 1 molecule acts similar to a telephone. It is one line of the telephone committing a conversation between itself and the receptor cell on either a T cell or other cell that needs recognition. The MHC class 2 molecules are distinctly restricted to immune cells. And so what you find is MHC class 1 receptors will express and present only intracellular molecules. So antigens that happen to be obtained from inside the cell will be loaded onto an MHC class 1 receptor. Antigens that are loaded into an MHC class 2 receptor are located from outside the cell. So they are picked up by phagocytosis or an endocytic pathway and then loaded into an MHC class 2 receptor. What we also know is that MHC class molecules are very important in recognition of self. So our immune cells must be able to recognize the same MHC molecules across species. This specificity is part of why donation of organs is highly controlled. Our cells, if they recognize these MHC molecules as not being self, in humans we call this molecule human leukocyte antigen. Human leukocyte antigen is how we do HLA typing. We do so before we perform donor transplants. The MHC class 1 molecule is going to present endogenous self-antigen. So endogenous meaning from within inside the cell, and it'll also present intracellular foreign antigen. So location, location, location. So what we see here, this is an example of an intracellular virus replicating inside our host cell. This intracellular virus will be processed and presented on an MHC class 1 molecule to a CD8 CTL killer T cell. This is necessary to regulate immune responses because our self-antigens must be put on a self-MHC and recognized by other proteins, because we don't want inappropriate responses to take place between a antigen-presenting cell and those expressing the MHC class 1 molecule. There's additional regulatory steps that must take place for a CD8 T cell to be activated. We'll discuss those in a bit. MHC class 2 molecules consist of B cells and macrophages and dendritic cells. These cells continue to produce these regulatory receptors that are necessary for the process of antigen presentation. Dendritic cells, if they phagocytose an extracellular microbe, they then can present that extracellular microbe in the MHC class 2 receptor. The MHC class 2 receptor is loaded with that microbe. That microbe is now called an epitope. It is a distinct identifier that is processed and placed into an MHC class II molecule. The CD4 T cells, those T helper cells, must become activated by the process of antigen presentation. So the phagocyte phagocytoses the bacteria is digested and broken down, then the phagocyte will use that bacterial component that is now called an epitope. It will be loaded into an MHC class II receptor and presented to a T helper cell. We then see that T helper cell will become activated. We'll cover the process of antigen presentation to a T helper cell again in a moment.
Antigens are processed differently, depending on whether they originate within or outside. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class I MHC molecules. The combination is then transported to the Golgi apparatus and from there to the plasma membrane. Foreign antigens presented on class I MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class II MHCs, and the two structures combine. The MHC class II antigen complex is transported to the plasma membrane. The displayed MHC class II antigen complex can stimulate other immune system cells to respond to the antigen.
So antigen processing depends on the location at which the antigen is encountered. If an antigen is encountered in the cytoplasm of the cell, then that antigen could be either self or a viral cell or an intracellular bacteria. Intracellular antigens are placed on MHC class I molecules and presented to CD8 and activated by helper cells. MHC class II molecules are extracellularly derived. So these are from outside of the cell, and they're picked up typically by endocytosis. This phagocytic pathway, they are then processed and placed on MHC class II molecules. Those MHC class II molecules then are used to present to other cells and activate the immune response. How do T cells recognize these molecules on the MHC receptors? So T cells recognize these molecules by binding the processed antigens, now known as epitopes, together with the MHC molecule. So the T cell receptor has a specificity not only for the antigen, but it has a specificity for the MHC receptor. B cells, however, can bind free antigens. You can see here that this is a B cell receptor, and the free antigen, shown here, can be picked up by the B cell receptor without the help of an antigen-presenting cell. Clonal selection theory is literally the idea that of all the 500 genes that we have, these are used to possess specific receptors. I have a simple analogy shown here at the bottom. Only circles will fit in the circle, only squares will fit in the square, and only triangles will fit in the triangle. This is true for our T cell receptors. Our T cell receptors have a lock and key interaction, and they do so between themselves and the MHC molecule. The same goes for B cells. B cells, even though they do not require the antigen to be processed, do have a distinct lock and key mechanism where they interact with high specificity to that antigen, and they do become more specific to the antigen after exposure. The development of T lymphocytes or B lymphocytes take place in the bone marrow. So both pre-lymphocytes for T cells and pre-B cells exist there in the bone marrow. B cells, however, will stay in the bone marrow. These will become naive B cells once they have become mature. T cells, however, the pre-T cell will move to the thymus, and the pre-T cell will become naive T cell, meaning not antigen experienced, expressing either a CD4 or a CD8 co-receptor. So this is a receptor that will work in coordination with the T cell receptor expressed on the T cell surface. After the maturation process is completed, B cells can move from the bone marrow to the spleen to become mature activated B cells. T cells and B cells will migrate to secondary lymphoid tissues when antigen has been encountered. This will then cause the T cells to become activated T cells, the B cells to be activated B cells, which are now called plasma B cells.
The immune system can produce antibodies against millions of different antigens. B cells, which are constantly produced from lymphoid stem cells, are found in the blood and lymphoid tissues. Each B cell can synthesize only one of the millions of possible antibodies and displays this antibody on its surface. When an antigen meets a B cell having a surface antibody of the proper specificity, it complexes with the antibody. B cells that are not activated do not develop further. Stimulation of only the B cells that carry antibodies that react with antigen is referred to as clonal selection. Following antigen-antibody interaction, antigen-antibody complexes aggregate on the surface of the cell. This is called capping. The antigen-antibody complexes are then internalized. The activated B cell swells and begins to divide rapidly, producing a B cell clone. The B cells then differentiate into plasma cells and memory cells. Plasma cells produce the specific antibody that provoked its formation, whereas memory cells remain in circulation but do not produce antibodies. Memory cells can become activated by a later challenge by the same antigen. Many antibody responses require signals from T helper cells. The T helper cell is stimulated by an interaction with an antigen-presenting cell such as a macrophage. The macrophage ingests the antigen, digests it, and presents it on the cell surface via a class II MHC. The macrophage then activates a T helper cell that carries a T cell receptor capable of recognizing the antigen on the class II MHC of the macrophage. The B cell presents the antigen on its surface using class II MHC. The activated T helper cell interacts with the B cell and secretes chemical signals called interleukins to stimulate the B cell to differentiate into a plasma cell and produce antibodies.
This video illustrates that both B cells and T cells require each other for activation. B cells must encounter antigen to be selected to become memory cells or plasma B cells. They later then can be activated by a secondary stimulus with antigen with the help of a T cell. Likewise, T cells need antigen-presenting cells, which do include B cells, to become activated. By activating one another together, they prevent inappropriate immune responses. All lymphocytes have the same receptor specificity, FOS. Specific B cell receptors can also be described as immunoglobulins. So globulin proteins are located in the plasma portion of our blood. Globular proteins are free-floating, and they have distinct genes that allow them to be of specific types. These immunoglobulins serve as both B cell receptors and free-floating antibodies in the blood. The antibody structure consists of two light chains, these outer regions that are smaller. They consist of two heavy chains, these larger regions in the center. And then they consist of variable regions on both sides, and these are the antigen binding sites, as well as conserved regions, which are located especially here in the Fc component. So Fc component is the consisting of the heavy chain that distinguishes what type of antibody you're looking at. I'll explain this a bit further. So the distinct and individual variable chains, these will change after antigen binding. This process is called affinity maturation. Depending on the type of antigen that has been experienced by this antibody, we then may also have a change in the Fc region where the heavy chains then undergo a process called isotype switching. We'll discuss this in a couple extra slides.
Antibodies are remarkably diverse. Each person can produce millions of different antibodies, each capable of reacting with a different epitope or antigenic determinant. Each antibody of the immunoglobulin G class, IgG, consists of 2 light chains and 2 heavy chains. The sequence of amino acids in the constant regions of all IgG antibodies is identical, whereas the sequence of amino acids in the variable regions differs, depending on the structure of the epitope.
Both B-cell and T-cell receptors undergo genetic rearrangements to increase diversity. This diversity is necessary to have specificity to all the potential microbes and self-proteins that the cells will recognize. Lymphocyte activation takes place with the antigen-presenting cell, or potentially the mature B cell, interacting with the helper CD4 T cell. So first of all, B cells, to become activated, they can pick up their own antigens. They can find these antigens at any site or location, but they need a second activation signal by a helper T cell, a CD4 expressing T cell, before they are fully mature and activated. This process takes place because it's a checks and balance. It prevents the B cell from picking up an antigen and responding inappropriately. So once a B cell has presented and shown the antigen to a helper T cell that has the same specificity, then the signals are sent back and forth between the 2 cells, and the B cell is now an activated B cell. Activated B cells will differentiate into plasma B cells, which will make antibody now, or memory B cells, which can be reserved for long-lived memory and make antibodies if this particular pathogen returns. So the way that B cells and T cells interact is the B cell first can pick up the microbe with its own B cell receptor. So these B cell receptors can pick up this microbe, and it waits until a T cell comes along of the same specificity. The T cell then will confirm that yes, this is a microbe that we should respond to, and as a consequence of these cell-to-cell interactions, the T cell will cause the B cell to become activated. Once the B cell is activated, it will differentiate into a plasma cell, which will make antibodies now, or a memory cell, which will remember the microbe for a later production of antibodies. The plasma cell will participate in the current immune response, secreting antibodies, which will coat the bacterial surface. The coating of this microbial surface will increase the rates of phagocytosis, will work with complement, and potentially facilitate the additional lysis and inflammatory cascade that are necessary to get rid of this microbe. T-cell receptors will also undergo genetic change to increase specificity for the antigen. In particular, most T cells possess an alpha-beta chain, shown here. The alpha-beta expressing T cells are that of our CD8 cytolytic T cells and our helper T cells that we'll discuss later. Gamma-delta T cells have much different responses than that of the alpha-beta T cells. T-cell maturation takes place in the thymus. The process of T-cell maturation in the thymus requires antigen presentation to take place in the thymus so that the T-cells can become antigen experienced. T-cell maturation is, in a sense, a first time that a T-cell is activated. Once a T-cell is activated, there is a co-receptor that is upregulated. It is called cluster of differentiation, CD, CD4, or CD8. T-cells will express one or the other, never both. These mature T cells, once they have been activated in the thymus, if there is an infection that requires their help, these T cells will then migrate to the lymphoid organs to be activated by antigen presentation. T cells become activated by the process of antigen presentation. Antigen presentation takes place when you have a phagocytic cell, like a dendritic cell, that will then consume a microbe. That microbe then is processed and presented on its cell surface in an MHC class II molecule to a helper T cell. Once the helper T cell uses the T cell receptor to recognize this foreign epitope, it then can become activated. Once the helper T cell is activated, it can secrete cytokines that tell other immune cells what type of infection needs to be cleared. CD4 T cells after being activated, will then differentiate into distinct helper T cell types. In some cases, helper T cells will differentiate into memory cells so that the pathogen, if it were to return later, it can remember it and immediately secrete cytokines. Other helper T cells will act at that point and become an activated helper T cell that will secrete cytokines to the immune cells within the site of infection. Regulatory T cells, these cells are very important to maintain immune homeostasis. They are upregulated at the conclusion of infection so that the immune cells do not overreact. They act as a fine balance between responding to the microbe and then causing autoimmunity. Regulatory T cells are critical for making sure that you maintain immune homeostasis. Helper CD4 Th17 cells are necessary to recruit neutrophils to the site of infection are important because they are the most abundant phagocyte. They're necessary to clear and immediately phagocytose any extracellular microbes. CD8 T cells respond distinctly to MHC class I molecules. If you recall, MHC class I molecules are most definitely expressed on all cell surfaces except for red blood cells. Because MHC class I molecules are expressed all throughout the body, mature CD8 T cells have to have a second signal for activation. These helper T cells make sure that the mature CD8 T cell responds appropriately to the antigen that is being presented. Because if mature T cells were to get inappropriately activated, it could cause severe tissue damage. These mature CD8 T cells can be memory cells that will then remember the pathogens later, and they can kill those infected cells if they return and happen to have an infection. Or they become cytolytic CD8 T cells now. These cells are going to lyse infected cells. Self-proteins are also expressed on MHC class I molecules. If mature CD8 T cells did not have a second layer of immune regulation added, they could accidentally become activated to self-proteins. This could result in an immunocompromised patient and lysis of healthy cells. The cytotoxic CD8 killer T cells kill their infected cells by producing two molecules, granzyme and perforin. Perforins will poke holes in the target cell, which will cause an osmolaric dysregulation, and then granzymes will enter into those areas and cause apoptosis. Killer T cells are necessary for things like bacteria-infected cells or virally infected cells. CD8 T cells recognize cancer cells because of a decreased quantity of MHC class I molecules expressed on their cell surface. How do cells know a microbe is foreign? It is by recognition of that antigen. Previously, we talked about antigens as pathogen-associated molecular patterns, but antigens can actually range in all sizes and shapes, and they're all antigens that are either recognized as self or not self. Antigens must have antigenicity, meaning they must have a foreignness, meaning not recognized in the body. They have to be of a certain size, shape, and they must be accessible for receptors to bind them. The characteristics of antigens are often broken down by phagocytes and processed into antigenic determinants. The antigenic determinants are epitopes. Epitopes are layered into the MHC class I or class II receptors, and they are then presented to the respective T cells. Antigens can have many antigenic determinants. A simpler example of this is yourself. So you could be recognized by your hands, you could be recognized by your face, you could potentially even be recognized by tattoos or bodily modifications that you may have that are distinctive from you and other individuals. These would be the antigenic determinants that are on your body. So our antigenic determinants are distinct to the individual. In this case, our antigenic determinants, or broken down epitopes, are distinct to the microbe.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes.
Each different antibody binds only to the correct epitope.
The immune system does not recognize haptens because they're just too small, or the quantity of information that they're providing is just not immunogenic enough. And so what happens, haptens often have to be complexed with a carrier molecule to be recognized by the immune system. The example shown here is a hapten that is injected into a mouse. This hapten does not elicit any antibody production because the hapten is just simply too small. The immune cells do not recognize the hapten, don't process the hapten, or present it to other immune cells. The hapten, however, if it is bound to a carrier molecule, it is now large enough and more antigenic. When it is more antigenic, then the T cells will become activated, B cells in turn will become activated, and will produce antibodies that can later then opsonize this particular hapten. There are distinct categories of antigens, one of which is that of a superantigen. Superantigens are potent T cell stimulators and were discussed in Chapter 18. These potent T cell stimulators bind the antigen receptor inappropriately, and as a consequence, there's an initiated T cell response, but it's not to the antigen of choice. It's actually only seeing part of the story. So as a consequence, you have a very overwhelming T cell response, but it is nonspecific. There's a video to explain this illustration in the next slide. Other types of special antigens are those of allergens. Allergens are antigens that provoke an allergic reaction. Autoantigens, this is when we have an autoreaction to our self molecules. This is unfortunate in the sense that it causes autoimmune diseases. It takes place when tolerance is inadequate. Tolerance is the ability of our immune cells to recognize self-proteins and elicit no response.
Antigens are normally processed by antigen-presenting cells and presented to T helper cells on class II MHCs. Only the T helper cells with T cell receptors capable of interacting with the antigen are stimulated to produce cytokines. The activated T helper cell stimulates only B cells that react with the same antigen, thereby limiting the response to the production of antibodies that were presented. Superantigens, such as the toxic shock syndrome toxin produced by Staphylococcus aureus, are not processed by antigen-presenting cells. Instead, they bind directly to the outer portion of an MHC class II antigen on antigen-presenting cells, and to the outer portion of the T cell receptor of T helper cells. Rather than binding to only the cells specific for that antigen, which represent about 1 in 10,000 T cells, the superantigen binds without antigen specificity to as many as 2% to 20% of the T cells. This results in stimulation of many T helper cells. The result is the release and activity of excessive amounts of interleukin-2, IL-2, and other cytokines that enter the bloodstream instead of only acting locally as they normally do. This leads to symptoms of fever, nausea, vomiting, diarrhea, and sometimes shock, with failure of many organ systems, circulation failure, and often death.
So as you can see, superantigens can be quite devastating for a person during an infection. The example shown here was toxic shock syndrome toxin. Streptococcus pyogenes also secretes various superantigens that have similar effects. The third line of immune defense is broken down in both T cell and B cell responses. The antigen-mediated immunity by B cells is considered the humoral immune response. Plasma B cells must be activated by cytokines secreted by T cells. Once this army has been produced, plasma B cells that will constantly secrete antibodies until this microbe is gone. The memory cells then will continue to persist within the body, protecting you from this microbe later. An example is chickenpox, which allows for immune protection throughout life. If the infection has passed, plasma cells will then undergo a contraction phase using apoptosis, which is programmed cell death. This allows that only the specialized memory cells will persist, and so essentially, if we're thinking about this in the war example, the army has now been discharged, its troops, you only leave a few people left in reserves. Antigen-antibody complexes are broken down into opsonization, neutralization, antibody tagging, and complement fixation. Opsonization is the coating of a microbe with either antibody or complement proteins and increasing the rates of phagocytosis. You can see opsonization shown here. Neutralization, however, is when antibodies have covered the coating of the microbial surface and it prevents the microbe from attaching to the host cell. If a microbe cannot attach, it cannot infect. Additionally, there's things such as antibody tagging, which will then increase the rates of phagocytosis. So this antibody Fc region can be bound by phagocytes, and now phagocytosis is going to increase. Complement fixation is when microbes are coated with antibodies and complement proteins, resulting in the membrane attack complex. The classes of immunoglobulins have distinctive functions. In particular, the first immunoglobulin to be made is IgM. IgM is the largest immunoglobulin. It is the pentamer, so it has 10 binding sites. The binding sites are the variable regions shown here. And so the 10 binding sites allow for a lot of interactions between this particular IgM molecule and different microbes. Once the microbe has been bound by the IgM molecule, affinity maturation is where the variable region of the antibody will modify and change to increase specificity for the antigen. Another component will take place, such as isotype switching. Isotype switching takes place in the Fc region. The Fc region will modify, and so IgM or IgG will be modified or class switched into IgA, IgE, or IgD. This is explained further on this slide. So IgM and IgG are produced first. Some molecules of immunoglobulin are necessary for different immune responses. In particular, if you need an IgA, IgD, or IgE molecule, they must undergo class switching. In other words, the Fc region of the antibody chain, this region shown here, must be modified from IgG to IgA. In this case, the classes of antibody change based on the necessity of antibody production. IgM being the first antibody produced, it can serve as a B cell receptor on all B cells. IgG, however, is produced by plasma cells, and it can be elicited in both the primary and secondary responses. The genetic rearrangements that take place for IgG to undergo class switching to either IgA, D, or E allow for specificity. IgA, for example, will circulate in the blood. It can actually be a dimer that produces antibodies during mucus and serous secretions. So this particular antibody is important in the gastrointestinal tract and the respiratory tract. IgD serves as a specific B cell receptor on B cells. IgE is involved in allergy responses and parasitic worm expulsion. IgE will work in concert with basophils, eosinophils, and mast cells to initiate degranulation that facilitate the process of allergies or facilitate the removal of parasitic worms. All antibodies in the IgA class have identical Fc regions, have identical antigen binding sites, have identical variable regions, are all monomers. The answer is A, have identical Fc regions because they are IgA. The primary response to an antigen is facilitated by first IgM. IgM, being the pentamer in the large molecule, is necessary for lots of variability to interact with microbial surfaces. IgM, for example, does take time before antigen exposure to be elicited. And so within this first encounter of antigen, or the first microbial encounter, there is a lag period of nearly 10 to 12 days before any antibodies are produced. Within this lag period, it's our first and second line of immune defense that we're relying on for protection. Eventually, IgM is made, and then shortly after IgM is made, we make IgG. These should both clear the microbe from our bodies. However, we do vaccinations to prevent the delay in this process, and instead what we see is that through vaccination we can result in a secondary response faster. So IgM no longer has a lag, and soon after IgM is produced, you have IgG produced in the secondary response. A secondary response is the second time that the immune cells have encountered the same antigen. So as a consequence of this, it can be achieved by natural infection or by vaccination. Regardless of that effect, due to the lack of a lag, our immune cells are able to rapidly clear the microbe and provide complete protection. There are various monoclonal antibodies that we use for treatment. So monoclonal antibodies are antibodies that are created in a lab that have a single specificity. We use them for various anti-cancer medications as well as for lymphomas. And so these are ways to supplement the immune response and help bolster that when we have inappropriate reactions. The lymphocyte responsible for finding and destroying cancerous cells is the antigen-presenting cell, the B cell, the cytotoxic CD8 T cell, or the T helper cell. The correct answer is the cytotoxic T cell. Immunities can be classified based on whether they are an active response or a passive response. Likewise, these can either be acquired naturally or through an artificial mechanism. Active immunity is when the immune cells are producing their own antibodies and their own activated cells. Passive immunity is when cells are provided pre-prepared antibodies for usage. As a consequence, active immunity is considered long-lasting. It takes time to create this memory, and so it is much more delayed than that of passive immunity. Passive immunity provide preformed antibodies, so there's no delay for making memory, but it acts immediately, but it only acts for a short period of time. Natural immunity, however, is our own natural immune system responding to a microbe. Natural immunity is obtained of normal life experiences, such as a child being exposed to chickenpox virus and obtaining chickenpox. Artificial immunity, however, is acquired through medical procedures such as vaccines. We now have a chickenpox vaccine, so many children never experience chickenpox. As a consequence, they are instead protected by artificial immunity. As we break these down, active immunity simply means that the microbe is responded to by our own immune cells, producing T cells and B cells that are necessary to get rid of it. Passive immunity, however, is preformed antibodies. Passive immunity can be preparing antibodies from mother to child, particularly in the natural venue. Active immunity, however, is when you are providing the antigen that the immune cells must themselves process, prepare antibodies and specificity to. Passive immunity, however, when it is artificial, is when we have taken immunoglobulins such as monoclonal antibodies and we're giving them to a sick patient to help them with their disease progress. We can provide passive immunity by immunotherapy mechanisms by using pre-prepared antibodies or immunoglobulins. This is one of the ways that we treated Ebola. So one of the main mechanisms and therapies against Ebola for efficacy was that of passive immunity. Patients who had previously been exposed to Ebola and survived, that were of the same blood group, provided blood donations to sick patients. This actually allowed for many patients to survive this most recent outbreak. Additional choices are shown here. So sera produced from horses, for example, are often used to treat botulism using an antitoxin, as well as spider or snake bites. So these antivenoms are literally pre-prepared antibodies. These antibodies are going to be able to act immediately and provide protection for a short period of time of 2 to 3 months. Vaccinations are artificial active immunity. We are inserting the antigenic components of a microbe into our bodies. These antigenic components will stimulate a primary response. If you recall, inflammation and those inflammatory cascades take place in our second line of immune defense. Vaccinations are actually going through our natural immune system. And so as a consequence, vaccinations although they're artificial, are still going through the active immune response. So as a consequence, we do have inflammation. We will have an inflammatory cascade that takes place as those antigens are being processed. This immune development is often very immediate, powerful, and sustained. The qualities of an effective vaccine, first and foremost, should not have adverse side effects or toxicity. We don't want to cause harm to our patients. Secondly, they need to be protective against exposure. So it should provide enough immune protection against the natural microbe at which you're vaccinating for. Additionally, it should stimulate both antibody and T-cell mediated responses. If it stimulates only one, it is not a protective vaccine. Finally, it should also have long-term lasting effects. If it does not produce memory cells, that is not a good vaccine. Vaccine preparation. Vaccines are prepared by either killed whole cells or inactivated viruses. They could be live or attenuated. They could be distinctly antigenic molecules, or they could be genetically engineered. Killed or inactivated vaccines consist of vaccines that are treated with formalin or completely deactivated with heat or other chemicals. As a consequence, these vaccines are dead. They possess antigenicity, meaning the surface proteins, but they are not alive. They require a larger dose, and often they require boosters to be effective. These vaccines will stimulate an immunity, but because the microbe will not be multiplying because it is dead, you have to give a larger dose to activate enough immune cells for efficacy. Live attenuated cells are live cells that can actively replicate. These cells then have the same antigenicity as the normal microbe, but what has changed is the microbe has had the antigenic factors that are causing damage or severe virulence removed. An example of this is the live attenuated influenza vaccine. The live attenuated influenza vaccine is a temperature-sensitive mutant. That means that the live attenuated vaccine that is given through the nasal cavity can only replicate in the nasal cavity. If it were to move toward the respiratory tract or the deep portions of the lungs where pneumonia is caused, so as a consequence of that, healthy patients are protected with that live attenuated vaccine because it can't replicate in a site that would cause disease. However, individuals who are immunocompromised are not recommended to have live attenuated vaccines due to the risk of active immune infection. Attenuated versus heat-killed vaccines. The advantages of these live protections is that they multiply, produce, and so they elicit a robust immune response. They confer a long-lasting protection, and they do so with fewer doses and fewer boosters. However, the disadvantages include special storage such as refrigeration, and could potentially be transmitted to other people or mutate back to a virulent strain. Antigenic molecules are acellular or subcellular vaccines, or just complete separated subunits or proteins. For example, a capsule, a surface protein, or a distinct exotoxin can be isolated. These can be isolated and vaccinated against to prevent disease. The exotoxins of diphtheria or tetanus are part of the reason the disease is so severe. So by vaccinating against the most severe virulence factor, these people are protected. Genetically engineered vaccines are when we actually insert distinct vectors into plasmids. Plasmids you may recognize from Chapter 4. Plasmids taken from bacteria, if you insert the gene for various proteins that are necessary for a vaccine, you can insert them into yeast or other cells that will then make that protein for you. The isolation of that protein then can be placed into a vaccine. If the capsule of a pathogen is being used as a vaccine, it would be called an acellular vaccine. The administration of vaccines sometimes require things called adjuvants. Adjuvants are necessary to increase the immunogenicity of a vaccine, especially those that are already dead or heat-killed. So our dead or heat-killed vaccines lack immunogenicity and have an adjuvant added to this particular vaccine component. What happens is as the immune system is processing the adjuvant, they also pick up the antigen that is present from the original vaccine. This then causes more side effects. Herd immunity is the idea that you have safety in numbers. So if we vaccinate large quantities of populations of people, the likelihood of that microbe surviving or persisting from person to person within that population goes down. You can see here that you have non-vaccinated individuals, and then in this case, if you put a sick person in that group of individuals who were not vaccinated, the entire group can become sick. However, if you were to have those healthy immunized individuals, they could reduce the spread of the infection across the population. This is exactly what we do annually with the flu vaccine. The flu vaccine is taken by lots of individuals, and they reduce the likelihood of harboring that particular pathogen and spreading it to others. So it is less likely that non-immunized people who are not able to get the vaccine are going to encounter the pathogen. There is a bit of controversy over vaccinations. In particular, there's recent studies that suggest that vaccinations supposedly cause autism. If you were to research this information, you would find that all the research has actually been debunked. However, because of the rise of autism in populations, there's a great movement to anti-vaccination campaigns. These anti-vaccination campaigns are resulting in new and recent outbreaks of measles at Disneyland, as well as other re-emerging diseases that we thought were once eradicated. Please review the learning checkpoint for these questions for this section of your exam.